to you?
Yes, you're audible.
Okay. Hello, everyone. Good afternoon. I welcome the experts, the members, students, farmers, entrepreneurs to this presentation. I am Dr. Aditi Sharma. I'm working as assistant professor in plant pathology under Dr. Yashwant Singh Parmar University of Horticulture and Forestry, Solan, which is situated in Himachal Pradesh. The topic of my presentation is current trends in the management of bacterial pathogens. So, as we know, plants are regularly challenged by many pathogenic microorganisms that reduce the quality and quantity of the produce and are major threat to food security. Among these pathogens, bacterial pathogens are also there. They are very destructive in nature and they are extremely small microorganisms. They are basically prokaryotic microorganisms. As we can see in the picture, bacterial structure is there. Bacteria are very,
Dr. Aditi, sorry to interrupt. Your screen sharing is not there. I mean, your presentation is not visible.
okay, madam. Wait, I'll see.
Yeah.
Now it's there, ma'am.
Yeah.
Okay, so I'll start once again. I welcome you all to this presentation. The topic of my presentation is current trends in the management of bacterial pathogens. First of all, we know plants are regularly challenged with different types of pathogenic microorganisms, and they are a major threat to food security. So, among different pathogens such as fungi, viruses, nematodes, bacteria are also there, and bacteria are very simple microscopic microorganisms. They are basically they are prokaryotic microorganisms with a simple structure. They are having different metabolic pathways. They are very destructive plant pathogens. But in case of bacteria, we know that the first bacteria to be known was bacillus, and the disease that caused was uh, it was anthrax disease in sheep, and the name of the bacteria is Bacillus anthracis. It was discovered by Robert Koch. This discovery was followed back by the discovery of the plant pathogenic bacteria, that is Erwinia amylovora. So bacteria are extremely small in size. They vary in shape and size. On the basis of cell wall structure, they are divided into two groups, that is gram positive and the gram negative ones. Basically, the structure of the cell wall, the composition of the cell wall is there. Therefore, they are divided into two different groups. And on the basis of size and shape, they are also divided. They generally reproduce by binary fusion, and unlike fungi and plants, they cannot synthesize their own food. They are dependent on decaying organic matter for their food. And when we talk about the bacterial diseases, so bacteria cause many diseases in economically important plants and is a threat to food security, as well as there are several bacteria that are of quarantine importance also. They spread from one plant to the another plant by the help of wind, as well as by the help of natural openings, by the help of wounds, as well as by the help of different vectors such as leafhoppers and psyllids. They cause major diseases in rice, pomegranate, wheat, and many solanaceous crops, as well as in cucurbits and in fruit crops also. Their typical symptoms include canker, leaf spots, water-soaked spots, and soft rots, etc. This figure here is depicting the general classification of bacteria. We know bacteria is a prokaryotic microorganism. They are divided into three different groups: the proteobacteria, firmicutes, where there are gram-positive bacteria, the actinobacteria also. So most of the gram-negative bacteria are pathogenic in nature. And there are several examples of gram-negative bacteria, such as Pseudomonas. This is one of the major pathogen of tomato and other crops. This causes bacterial speck. You can see in the picture also The other bacterium known as agrobacterium, this causes crown gall disease. Irvinia, very important pathogen, causes black leg in potato. And
Dr. Aditi?
instead of this, bacteria can also get entry by the help of wounds and by the help of root hairs. Bacteria, as far as the virulence of the bacteria is concerned, they produce several cell wall degrading enzymes that can degrade the cell wall of the plants. They can produce phytotoxins that can modulate the cell membrane and that can modulate the cell structure that kills the plasma inside the cell. They can also modulate the plant hormones. Bacteria has several virulence genes, and in case of agrobacterium, that bacteria can transfer the tDNA to get entry inside the plant host system. There are several type secretion systems like type 1 secretion system, 2, 3, 4, and many more. By that, these secretion systems, bacteria can also get bacteria take the help of these secretion systems for their virulence processes. At the plant microbe level, we know that the effectors or the virulence factors secreted by the bacteria or other pathogens, such as the extracellular enzymes, the toxins, they are secreted in the bacterial cell, and they get entry inside the plant by the help of these secretion systems. Among them, type 3 secretion system of bacteria is very important because this secretion system acts as injection, and the effector proteins get entry inside the plant system. So instead of this, in case of plant, when we talk about plant, when plant senses the foreign components coming inside the plant tissues or cells, plant recognizes these effector proteins and initiates the defense mechanisms that is present in the resistant plants. So this plant microbe interaction goes well if there is a virulent pathogen and a resistant host. According to molecular plant pathology, there was one review that has focused or that has addressed top 10 major bacterial pathogens that infect plants. Among them, these are the list of different bacterial pathogens such as Pseudomonas syringae, Ralstonia, Agrobacterium. Pseudomonas syringae is a very important pathogen. This causes speck disease in tomato also, and many more crops that are of economic importance. Ralstonia solanacearum, as we know, causes wilt disease. This belongs to the family Burkholdiaceae. This is a rod-shaped bacteria and causes many wilt diseases in different solanaceous crops. This blocks the xylem vessels, and the transport of water and minerals is reduced, and finally the plant wilts. It loses its turgidity and plant wilts. So this is also an important pathogen. When we talk about Agrobacterium tumefaciens, this pathogen is very smart because it has the ability to transform the host or the plant cells. It transfer DNA, we call it as tDNA, into the plant genome, and then thereby induces the overgrowth of the plant by producing certain enzymes such as auxin and cytokine. So this is a bacteria of quarantine importance also. This causes crown gall disease, which is known as a tumor in case of plants. So this causes tumor or crown gall in apple, walnut, stone, and pome fruits, as well as in rose and grapevine, etc. Xanthomnas is also an important bacterial genera. This causes bacterial blight of rice, bacterial blight of rice, and this pathogen is very important, and in case of quarantine, it has quarantine importance also. So Xanthomonas produces a pigment known as xanthin. This pigment and the extracellular polysaccharide in the Xanthomonas, they are responsible for virulence in various hosts. Instead of this, there is Erwinia mylovora. You can see the cause of fair blight of apple and pear. This is an important pathogen. It belongs to the family Enterobacteriaceae. And then Xylella fastidiosa, we have seen that in the recent years, oak wilt was there. Oak wilt and dieback. This was the major pathogen responsible for dieback and oak wilt. So Xylella fastidiosa is also very important bacterial pathogen. This is a gram-negative bacterium. And there are two other bacterial genera. They are also very important, such as Dictea and Lactobacterium. These genera are responsible for rotting symptoms in plants as well as in fruits and vegetables. The rotting is just because the bacteria produces certain enzymes that degrades the cell wall components, and therefore maceration and softening of the tissues occurs, and finally the plant and the fruit or vegetable rots. So these are some examples of top 10 major bacterial pathogens that cause so many diseases in different economically important plants. So this is about bacteria, how bacteria are virulent, how pathogenesis occurs in bacteria, what are the virulence factors responsible for a successful penetration inside the host plants. These all are the factors that I have discussed. Now I will focus on the topic given to me, that is bacterial disease management. So we have seen that bacteria are becoming so much important for us, because they are causing very severe losses to our fruit crops as well as to our agriculture crops. So management of bacteria is very tedious task. It requires a thorough knowledge of the pathosystem. It requires a thorough knowledge of the plant as well as the bacteria. So earlier management approaches were based on integrated approach, such as use of cultural control practices, in which we have gone for crop rotation, soil amendments, fertilization, etc. And then we have biological control practices. They have also adopted chemical control by the help of copper-based fungicides, and then an era of antibiotics started, where antibiotics were extensively used by different farmers and workers throughout the globe to manage these bacterial pathogens. But with the advent these copper-based chemical compounds and antibiotics, the pathogen or the bacteria became resistant to these antibiotics, and finally bacteria developed antibiotic resistant genes so as to overcome the effect of antibiotics. This is a picture that is showing that how antibiotics are how antibiotics are important, and how antibiotics get entry inside the environment. Antibiotic spray generally this antibiotic spray drifts off the target, and animals feed on the antibiotics contaminated grasses, as well as insects feed on the antibiotic contaminated leaves. Finally, these antibiotics get entry into the gut microbiota of the animals and get entry inside the soil, and this influences the soil microbiota or microbiome, as well as these antibiotics are influencing or are very harmful for phyllosphere microbiome also. So how the resistance against these antibiotics develop in the bacteria? Basically, this is a process known as horizontal gene transfer. In this, Resistant genes are encoded by antibiotic producing organisms, and they are transferred to the non-resistant ones. There may be acquisition of existing genes from the environmental microbiome. There may be spontaneous mutations in the bacteria, so they became antibiotic resistant. There are several examples of antibiotic resistance, and this is a very major very major component where we have seen streptomycin resistant genes in many plant pathogenic bacteria, that is Erwinia, as well as Pseudomonas, Xanthomonas, and many other bacterial pathogens. They have acquired antibiotic resistance from a non-pathogenic ones. So this is a major fact where antibiotics are not working now, and they are expensive too. So we are shifting our management practices to a way find a way where we can get eco-friendly management methods also. So the current approaches in management of bacterial pathogens involves many different methods. I will discuss them one by one. First is our bioactive compounds. The bioactive compounds are working well against bacterial pathogens. What are the bioactive compounds?
Dr. Aditi, Dr.
They
Aditi, are
are
you with us?
well as from the bioactive compounds, their composition varies. They can be salicylic acid components, they can be flavonoids, terpenes, proteins. Different bioactive compounds, phenolic compounds are there that are having antimicrobial activities, not only against bacteria, but also against viruses, also against fungi. Most of them are being used against fungi on large scale or commercial scale. And but now the studies are there, the research is focusing on the advancements where bioactive compounds can be used against bacterial pathogens also. One more fact of bioactive compounds is that they can be synthesized from different plant families, and plants are also they are the biofactories of these antimicrobial bioactive compounds. So this table is depicting the different studies that shows or that address the effect of these bioactive compounds against bacteria, such as essential oils. Essential oils they are synthesized from a plant source or animal source or a marine source, but they are effective against Clavibacter michiganensis. They are effective against Pseudomonas. Similarly, phenolic compounds are there, flavonoid compounds are there that can be synthesized from different plant families, and they are effective against Agrobacterium tumefaciens, Erythromycin vora, and Pectobacterium. There are several alkaloids also. They are able to target Ralstonia, Solanacearum, Erwinia, Pectobacterium, and Burkholderia, etc. So these are the studies which shows that bioactive compounds are now being used against these different phytopathogenic bacteria, and they are showing significant results against these bacteria. The second or the other important technique is bacteriophage therapy for inactivation of phytopathogenic bacteria. So what are bacteriophages? Basically, bacteriophage is a virus that is able to infect bacteria. So infection of bacteria, and this is a big process where a virus gets attached to the bacterial surface. It gets penetrated inside the bacterial cell, and then replicates inside the bacterial cell. And after replicating, making its progeny, it synthesizes its own machinery inside the bacterial cells, and after that the progeny infects the another progeny infects other bacteria in the vicinity of the viruses. So this is a very good technique. It is also a environment friendly technique which can produce exponential number of viruses that can infect bacteria and can be actively used for the management of bacteria. There are several examples of bacteriophage technique in the recent past. From the year 2012, I have made the table to 2021. There are so many examples. Other than this, you can check on the internet, and these bacteriophages are working very well because they are making the formulation of bacteriophages with certain carriers, and some resistant bacteria are also, they are using some resistant bacteria also for making the formulation of these phages, and then delivery of these phages to the to control the different bacterial diseases such as wilt diseases, canker, and leaf spots, etc. These phages belong to different families and different families, but generally Podoviridae and Myoviridae families are very important, and the phages present in these families are working well against different bacterial pathogens. So when one phage is effective, they are also making phage cocktails by using two different family phages so as to protect the different bacteria against the protect different plants against these bacterial pathogens. So you can see the table. There are different types of bacteriophages that are able to control the wilt pathogens, the pathogens or the bacteria that cause leaf blight and that cause canker also. So bacteriophage technique is also an important one. Other technique that is introduced is nanobiopesticides or nanoparticles. As we know, nanoparticles are very small particles. They range from 1 to 100 nanometer in size. They are so small, and they vary in composition also. So due to their ultra small size, they are having the potential, or they are highly reactive in nature, and they have great potential to be used in the diagnosis of bacteria as well as diagnosis of fungi and different pathogens and microbes, and they can be actively used for the management of bacteria. The nanoparticles, we have another fact that there is another word, we call it as green nanoparticles, where we add some plant material to the nanoparticle so that it becomes eco-friendly in nature, because many plants have antimicrobial activities. So we are amalgamating these two components together to make eco-friendly green nanoparticles. And the nanoparticles can also be integrated with other biological materials for detection and for the management purpose also. This is the table where I have added different examples or references regarding the use of nanoparticles. Silver nanoparticles, copper nanoparticles, iron-based, zinc oxide-based nanoparticles are now being used against different bacterial species or genera, and they are working very well because the delivery system of these nanoparticles is easy and the synthesis process is also easy. So silver nanoparticles have been widely used against bacterial wilt diseases. Copper oxide nanoparticles are also used against Ralstonia solanacearum, and many more phytopathogenic bacteria has been controlled by the use of these nanoparticles. Another approach which is being used nowadays is quorum quenching mediated management. So as we know, when the battle is there between bacteria and the plant, so bacteria uses its cell-to-cell communication machinery that is known as quorum sensing machinery, and bacteria communicate with each other, make the biofilm, and became active as virulent pathogens. So in response to this, plants often have some antimicrobial compounds that inhibits the action of quorum sensing signals, and this phenomena is known as quorum quenching. So quorum quenching is basically the switching off of the quorum sensing signals. This can happen by the help of inhibition of synthesis of quorum sensing signal in bacteria. And quorum quenching can also be, it involves the inhibition of sensing of those quorum sensing signals. It involves the degradation of quorum sensing molecules. So there are several examples where we have seen that quorum quenching has been implemented so as to manage the bacterial pathogens. But there are different organisms which are involved in this quorum quenching mechanism, such as Rhodococcus, Ralstonia, Klebsiella, Pseudomonas aeruginosa, and some plant extracts also. Basically, quorum sensing mechanism involves acyl homoserine lactones, and so as to defeat these molecules, quorum quenching bacteria produce certain enzymes that can block the signal sensing, and then uh, that can synthesize some structural components that are analogous to this acyl homoserine lactones. These quorum quenching organisms can also produce diffusible signal factors. They degrade these diffusible signals and modify these signals so that the bacteria cannot infect the plants. So the phytopathogens treated by the help of this quorum quenching mechanisms are Pseudomonas, Agrobacterium, Erwinia, Xanthomonas oryzae, and Ralstonia solanacearum, etc. There are several references in Google Scholar which advocates the use of this technology, where we have also transgenic bacteria that are acting as quorum quenching organisms. They are modifying the cell wall machinery. They are modifying the acyl homoserine lactone in bacteria, and they are inhibiting the action of these molecules so as to reduce the pathogen attack. There are several bacteria such as streptomyces. Uh, they are able to switch off the mechanism of biofilm formation in Ralstonia, and there are several more examples also where quorum quenching has been seen successful under in vitro conditions. Then there is genome editing approach. Genome editing approach is also very important, and nowadays CRISPR-Cas mechanism is working very well. CRISPR-Cas is basically a DNA sequence, and this was discovered in bacteria and archaea, and bacteria utilizes this CRISPR system when it is attacked by a virus. So it recognizes the attack of a virus, and then it switch off, or it cuts the genes that are involved in virulence processes. So this is a good mechanism. This is a good approach to be used as a management approach against different bacteria. And genome editing techniques are successfully employed in several economically important crops such as rice, tomato, citrus, apple, grapevine, banana, etc. So these are several examples where genome editing technique of CRISPR-Cas9 has been utilized or implemented well against different bacteria. So we have seen that I have made the table from 2015 to 2021. There are so many examples where this system has been adopted against bacteria. So now I will conclude the presentation. As we know, there are bacteria are these small microscopic entities or prokaryotic microorganisms. Their management is difficult because they are getting antibiotic resistance, and they are very minute. They are not able, some of them are not able to be cultured. So this is a tedious task to manage these bacterial pathogens, but the current management approaches are focusing on environment friendly methods so as to manage these bacteria. There are I have discussed bioactive compounds, I have discussed bacteriophages, green nanoparticles. These techniques are very useful, and these methods are being employed by different workers throughout the globe. And uh, and now CRISPR-Cas technology, these all techniques are having in vitro studies. Some of them have been utilized under field conditions also. But besides this, mm, there are so many factors, there are so many limitations of these that need to be addressed, such as the method of delivery of these molecules under field conditions, how they will work, how the consumers will accept these new molecules, how the public will accept the genome edited crops and other mechanisms or other management tactics. So all these limitations are also there, but we have to focus on the better side. We have to focus on the environment friendly methods that can be implemented to manage these bacterial pathogens. As science has no limits, somewhere something incredible is waiting to be known. Therefore, we should keep on researching, we should keep on trying new methods for the management, so as to manage these bacterial as well as other microscopic organisms. Thank you. Thank you. Have a nice day.
Thank you, Dr. Aditi Sharma, for a very detailed presentation. I'll now take up the questions. So the very first question is, what is the difference between virus and bacteria diseases in plants?
Basically, in plants, first of all, we should know what is virus and what is bacteria. Bacteria are the prokaryotic microorganisms having a cell structure, cell wall, and all these organelles present in bacteria. But viruses are something else. Viruses are made up of either DNA or RNA, and they consist of a protein coat. So the structures of both of them are different, as well as the mechanism of action on the plant is also different. If we see bacterial diseases, the spots are something different. In case of viruses, they basically they cause chlorotic type of spots, or you can see the mosaic type of pattern in viruses. If we talk about bacterial disease, you can identify bacterial disease on the basis of if there will be bacterial ooze coming from out from the plant. But in case of viral diseases, there will be no such ooze. You can and viruses are systemic in nature. The plant will be affected from root to the top systemically. But in bacterial diseases, you can see localized infections such as leaf spots, such as wilt is a systemic infection of bacteria, but you can see leaf spot, and I have told about crown gall, that is confined to crown and root portion. So likewise, we can differentiate between a bacterial disease and a viral disease, but first of all, we should know the difference between these two pathogens.
Okay.
Thank you.
Dr. Aditi, you can stop sharing the screen so that we can see your full video.
Okay, madam.
You can stop it, so it'll yeah.
Okay, ma'am, fine.
Yeah, it's fine. Thank you. And I think the baby is crying behind you. If you want to take a break, you can attend to her.
No, it's
No
okay.
problem.
It's okay. I'm sorry for that. Actually, he was
That's fine.
sleeping.
We can understand. So yeah, next question is, at the broad level, what can farmers do to avoid bacterial infection in their crops?
Okay, at broad level. First of all, we should think about planting material. Whatever, if you are collecting seed from somewhere, or if you are collecting some planting material, it should be free from bacteria, or it should be free from any pathogenic agent. our soil should be free from pathogenic microorganisms. You should go for crop rotation year to year, so that the inoculum cannot get multiplied, or it can be the multiplication of the inoculum year to year can be stopped. So all the I think management is not just a one tool. We cannot say that by using biocontrol agents you can manage, by using chemicals you can manage. You have to keep watching your field extensively, because you have to go for cultural control also. You, if you see anything, any plant infected, you have to cut it, you have to uproot it, and you have to you have to go for different cultural practices also. This is not a one, there is no one, I don't think that only one way is there, because they are very smart pathogens, and we have to be keen observer to manage them. We can on large scale, first of all, if you are just, this is my answer, if you want to make sure that
Dr. Aditi, we lost your voice. Hello, Dr. Aditi.
madam, I'm not
Yeah. Uh, can you hear me now?
audible, right? Yes, madam.
Yeah, you can hear me, right?
Yes, madam.
Okay. So I'll take up the next question now. At which stage of cultivation these pathogens start to grow?
Pathogens, actually, this depends on the plant, what type of plant it is, and what type of soil you are having. Pathogens continue to grow. There is only one overwintering. They can overwinter when there is unfavorable weather for them. Otherwise, they will continue to grow. So it will depend on the type of pathogen, type of soil, and the host plant in which the disease has to occur.
Okay, and which is unfavorable season for them or stage for them?
Okay, madam, unfavorable means if they are not getting the proper host or the proper pH, proper temperature, it will be unfavorable for them.
Okay, yeah. So next question is, please let us know a little about your education background and how you got interest in this topic of pathogens.
I have done PhD in plant pathology, MSc PhD both in plant pathology. Then I have worked on agrobacterium, which causes crown gall disease in stone fruits. I have worked on peach crown gall. So I have worked on bacteria, and I have interest in bacteriology. That's why I have chosen this topic. There was very few clues about bacterial disease management. You can find only few reviews, few research papers based on this management. So I have tried to club all the management tactics that has been employed, because I have to make it very convenient for all of us, so as to know how bacterial pathogens are managed in the globe. So
Okay.
that's why I'm having interest in bacteriology.
Okay. And next question is, do you provide any consultancy service or any guidance to the farmers?
When there is any WhatsApp message or any call from farmers point, then I can handle that. But for that you should have my number. For that you should tell me before.
Okay. I mean to say, any formal consultancy service you're providing?
Not yet, madam, not yet, because I have not that much experience yet. I am just at this stage of learning. So have just started my job as assistant professor in that university. So later on, I will be focusing on that.
Okay. Okay. And next question is, in terms of research on pathogens, what are some of the current or latest areas where research is going on?
Ma'am, I am not getting your question.
I'll repeat it 1 second. Yeah. What are the in terms of research on pathogens, what are some of the current or latest areas where research is going on?
Now we should say the latest area is omics, metabolomics, proteomics, genomics. This is metabolomics is now emerging as a science nowadays.
Okay. And where this research is going on? Any idea?
Maybe in IRI, New Delhi, in IITs, everywhere throughout the world. This is the new and instead of this, I think sensors are also important. Everyone is focusing on sensors, image sensors, RGB sensors, hydrothermal sensors for the detection of these pathogens and other microorganisms.
Okay. Okay. Okay, Dr. Aditi Sharma, I don't see any further questions from our participants. So now we have come to end of question round. Agricultureinformation.com likes to thank Ms. Dr. Aditi Sharma for a very detailed presentation and answering all the questions in detail. And we also like to thank all the participants for joining this meeting. The meeting will now be closed.
Thank you, madam. Thank you so much. Thanks to all participants from my side.
You're most welcome.